Hello everyone and welcome to the new session of the lecture series, and today we'll be discussing all the NEET previous year questions from the chapter Plant Growth and Development. So let's start with the first question. So here they are asking that the Avena curvature experiment is used for bioassay of so Avena, what is Avena? Avena is oat. Alright, so the options are abscisic acid ABA, second option is gibberellic acid, third option is indole acetic acid, which is an auxin. It is a natural auxin. Alright, then last option is the ethylene, which is a gaseous hormone. So let's discuss little bit background of this Avena curvature experiment. Alright, so FW went, he isolated auxins, from the oat seedling. He was working on oats. Alright, so what he did, suppose in this experiment, if this is an Avena coleoptile, alright, so auxins are produced in the tip. This is the tip region. Auxins are denoted by this red mark. So auxins are produced in the tip of the coleoptile, or you can say it's produced in the shoot tips. So next what he did, he excised this. He decapitated, he removed the shoot tip. Remove the shoot tip. And then what? What was the next step? This, this cut shoot tip was kept on a agar block. Alright, now this removal of the shoot tip is known as decapitation. Now what happened? Auxins from here migrated to the agar block. Auxins were initially present in the shoot tip. When the shoot tip was kept on the agar block, then auxin diffused in the agar block. Here you can write that the auxin diffuses into agar block. Alright, then what happened? This agar block was kept on the Avena coleoptile, which was decapitated on the one side. It was kept here. And then it was illuminated by unilateral light. Light from only one direction. Then what was observed when it was illuminated by unilateral light? Again, there was a curvature. There was bending of this coleoptile towards the light. Why? Because again, auxins was present in the agar block. This auxin diffuses into the coleoptile here. And a curvature is observed when unilateral light source is given to these coleoptile. And then the angle of curvature was measured. So this is known as agar block experiment. Agar block experiment. Simply you can say that this experiment, what does it do? It measure the angle of curvature of the coleoptile. Okay, after the agar block was placed on the decapitated coleoptile. Decapitated coleoptile, okay, on one side. Okay, so now this experiment was done to detect the presence of indole acetic acid, which is an auxin. All right. So what should be the answer? Answer is option C, indole acetic acid. Now coming to the next question, which is fruit and leaf drop at early stages can be prevented by the application of, so means here they're talking about abscission. Okay, abscission. And what is abscission? Detachment or the falling off of the leaf, fruits or flowers. Alright, so options are ethylene, then we have auxins, gibberellic acid, and fourth option is cytokinin. So here they are asking that the abscission at early stages can be prevented by the application of, so what should be the answer? Answer is option B, auxins. So what auxins have? Okay, it prevents abscission in young leaves and fruits. Okay, young leaves and fruits. But what does it do in mature leaves and fruits? It promotes abscission in older or mature leaves. Alright. So option B is the correct option, auxins. Okay. If you talk about the role of other phytohormones, ethylene is also known as the ripening hormone because it helps in the ripening process. Alright, gibberellic acid has several roles. Okay, malting, bolting. It also promotes seed germination. Okay, so you can write promote seed germination. Okay, bolting also, which is increase in the internode length just prior to flowering. And then we have malting process, speed up the malting process in brewing industry. Again, prevent abscission and all and everything. Okay, and cytokinin major role is delay of the senescence, and it also helps in the cell division. Okay. Now coming to the next question, which is what is the site of perception of photoperiod necessary for induction of flowering in plants? Alright. So they are talking about flowering. Okay. Means they are talking about the phototropism, photoperiodism. Alright. So options are lateral buds. Second option is pulvinus. Then we have shoot apex, and last option is leaves. Alright. So now we have already know that site of perception of photoperiod. Photoperiodism they are saying. So what should be the answer? Answer is option D, leaves. Alright. Site of perception is leaves. And what happens in leaves? Which hormone is produced? So it has been hypothesized that a floral hormone is produced. Okay, so this floral hormone is known as florigen. And what does, they are produced in the, so this hormone is produced in the leaves. And what type of hormone is this? It is a proteinaceous hormone. Alright, so this hormone is produced in the leaf, and then what happens? It is transported to the shoot apical meristem. Alright, and what happens? There is flowering then. Flowering is observed then. Or there is induction of flowering. Okay. Now coming to the next question, which is, it takes very long time for pineapple plants to produce flowers. Okay. Now which combination of the phytohormones can be applied to artificially induce flowering in pineapple plants throughout the year to increase the yield? Okay. So what does farmers do? They artificially induce flowering in pineapples. Okay. And induction of this female flower or early induction of the female flower leads to increase in the yield. Okay. So options are, so these are the phytohormones. First option is auxin and ethylene. Second option is gibberellin and cytokinin. Third option is gibberellin and abscisic acid. And fourth option is cytokinin and abscisic acid. Okay. Now we know that auxins, alright, they promote flowering in pineapple. Alright. And another hormone, ethylene. This also induces female flowers. Okay. And induction of female flowers, leads to increase in the yield. Alright. So what should be the answer? Auxin and ethylene. The next question is, which of the following is not an inhibitory substance governing seed dormancy? Alright, seed dormancy. What is seed dormancy? Temporary inhibition of the physiological responses of the seed. Alright, physiological response of the seed. And what happens when the conditions are adequate? Okay, then again seed germination can resume. Okay. Now options are abscisic acid, which is a growth inhibitor. Okay, it promotes seed dormancy and bud dormancy. That's why it is known as a growth inhibitor. Phenolic acid. Okay, phenolic acid is a chemical inhibitor. Okay, it also inhibits seed germination. Again, para-ascorbic acid. This is also a chemical inhibitor. Then last option is gibberellic acid. Gibberellic acid, we know it breaks the seed dormancy. Alright. It also promotes seed germination. Okay, so now the question is, which of the following is not an inhibitory substance? Okay, so gibberellic acid is not an inhibitory substance. So option D is correct. The next question is, name the plant growth regulator, or you can say hormone. Okay, name a plant growth hormone which, upon spraying on the sugarcane crop, increases the length of the stem, thus increasing the yield of the sugarcane crop. Okay, so what happens? Let's study the role of gibberellin first. So gibberellin, okay, it enhances the internode length, okay, or it increases the internode length, or you can say if the internode length increases, then the length of the stem also increases. You can say length of the stem increases or length of the main axis increases. Alright. So sugarcane, okay, if the length of the stem increases, then the yield will also increase. Alright. So options are gibberellin, ethylene, abscisic acid, and cytokinin. So we have already talked about gibberellin. Ethylene we know is a ripening hormone. Main function is ripening. Okay. Cytokinin against cell division and delay in senescence. Abscisic acid is a growth inhibitor. So what should be the answer? Answer is option A, gibberellin. The next question is, which of the following is not the function of auxin? Not the function they are saying. Alright. First is stoppage of the cambial activity. Second is initiation of new roots. Third is inhibition of root growth. And fourth is production of ethylene. Alright. So auxins we know are involved in the initiation of roots. Okay. They are involved in initiation of root, production of ethylene, stoppage of cambial activity. So this is not the role of auxin. Auxin doesn't inhibit root growth. Instead, it promotes the root growth. Alright. So what should be the correct answer? Option C. Auxins are not involved in inhibition of the root growth. Now next question is the site, okay, of perception of light in plants during photoperiodism. Alright. So options are leaf, shoot apex, stem, and axillary bud. Okay, we have done a similar type of question before. If we talk about photoperiodism, the site of perception is always leaf. Okay, so option A is correct. The next question is, plant follow different pathways in response to environment or phases of life to form different kind of structures. Now this phenomena or ability is called, options are maturity, elasticity, flexibility, or plasticity. Okay. So maturity, what happens at maturity? Mature cells are formed. Okay, and these mature cells, what do they have? They have some specialized roles or functions. Okay, but they are asking about the pathway in response to environment and phases of life. Okay, which forms different kind of structures. So this is not true. Second is elasticity. What is elasticity? Ability to regain that shape. Okay. It is the ability of any object or the material to, okay, regain the original shape. This is known as regain the original shape, okay, after stretching or after compression. Okay, what is flexibility? Flexibility is the ability to change according to the situation. Okay, ability to change according to the situation. Okay, Now let's talk about plasticity. Again, plasticity, what is the English meaning? Ability to mold or the quality to mold. Okay. In biology, how plasticity is defined? Okay. When one organism shows two different phenotypes. Two different phenotypes or two different forms. Okay. Depending on either the environment. Alright. Or it can be developmental. Okay. At different phases of life. Okay. Developmental means at different phases of life. Alright. Now, so what should be the answer? Answer is plasticity. Option D. Plasticity, what happens? The organism can have two different forms or shape depending on either the environment or the developmental stage. Okay. Example of environmental plasticity, okay, is seen in buttercup. Okay. In buttercup, what happens? The cells present in the air, okay, or developed in the air or terrestrial, they are lobed, okay, and they are large. Whereas, okay, some are produced in air and some are floating leaves or they are developed in the water. So leaves developed in water are dissected. Okay, and leaves developed in the air are lobed and broad. Alright. Similarly, developmental plasticity is seen in larkspur. Okay, cotton. And then we have coriander. Alright, in this, the juvenile leaves are different from the mature leaves. They are different in phenotype. They are different from mature leaves. Alright, so plasticity is the answer. The next question is, which one of the following plants does not show plasticity? Okay, so coriander show developmental plasticity, cotton also show developmental plasticity, buttercup show environmental plasticity. Alright. Cotton show developmental plasticity, coriander also developmental plasticity. So option left is maize. So maize is the correct answer here. The next question is, production of cucumber has increased manifold in the recent year, they are saying. Now they are saying the application of which one of the following phytohormones has resulted in the increased yield, as the phytohormone is known to produce female flowers in the plant. Okay. So hormone responsible for production of female flowers is ethylene. Okay. Gibberellins have some different roles. We have already discussed. Cytokinins also have some different roles. Abscisic acid is a growth inhibitor and a stress hormone. So answer is option B, ethylene. Ethylene, it enhances the production of the female flowers. It has been shown that female flowers in cucumber, alright, an increase in the female flower lead to the increase in the yield. Alright. The next question is, the gaseous plant growth regulator or plant hormone is used in plants to, first is, which is the gaseous plant hormone? It is ethylene. Okay. First option is promote root growth and root hair formation to increase the absorption surface. Alright, we know if the root growth and the root hair are extensively formed, or the growth of the root and root hairs are enhanced, then it will increase the surface area for absorption of the water and nutrients. Alright, so this is correct. Okay. Now, second option is help overcome the apical dominance, which is not correct. Third option is kill dicotyledonous weed in the fields. This is also not correct because 2,4-D. Okay, 2,4-dichlorophenoxyacetic acid, which is a herbicide. It has herbicidal activity. Okay, so this is used for the preparation of the weed-free lawns by the gardeners. Or weed-free gardens, you can say, or lawns. Alright, because it kills specifically the dicotyledonous weed and doesn't affect the monocotyledonous plant. The fourth option is speed up the malting process. This is again not true because for speeding up the malting process, gibberellic acids are required. Gibberellins. Okay, so what should be the answer? A. The gaseous plant hormone, which is ethylene, promote root growth and the root hair formation. Now, next question is, in tissue culture experiments, leaf mesophyll cells are put in a culture medium to form the callus. Okay, now this phenomena they are saying it's known as. So what is happening here? They have used the leaf mesophyll cells. Now what are mesophyll cells? These are differentiated cells. Okay, these cells are already differentiated, means they are matured cells. Okay, they have some specific functions. What is the function? Okay, they contain chloroplast, lot of chloroplast. Okay, function is photosynthetic. They are photosynthetic cells, helps in photosynthesis. Alright, now what happens if we want to induce callus from the mesophyll cells? Okay, so these were already differentiated. Then what is callus? Callus is mass of unspecialized cells. Okay, unspecialized or undifferentiated cells means they don't have any specific characteristic or function. Alright, so what happens? How callus will form? Now from differentiated cells, undifferentiated cells are produced, and this phenomenon is known as de-differentiation. Okay. So the options are differentiation, okay, which is not true because differentiation leads to the formation of mature cells or differentiated cells. But here callus is formed which is undifferentiated. Second option is de-differentiation. Okay, so this option is correct. Third option is development. So this is also not true because development is what? All the stages from the germination of the seed to the senescence. From germination to the senescence. Okay. Now last option is senescence, which is known as biological aging. Alright, so what should be the answer? Answer is option B, de-differentiation. The next question is spraying of which of the following phytohormone on juvenile conifers help hastening the maturity period that leads to the early seed production. Okay, so seed production is achieved during the reproductive stage, okay, or when the plants become mature. So what happens? How can we hasten the maturity period? By application of which hormone? First is indole butyric acid, which is a natural auxin. Okay, so this is not true because it is not involved in hastening the maturity process. Second option is gibberellic acid. third option is zeatin, which is again a cytokinin. So it doesn't have any role in hastening the maturity period of the plant. Fourth option is abscisic acid, which is a growth inhibitor and a stress hormone. Alright, so what should be the correct answer? Gibberellins. Gibberellic acid is involved in hastening the maturity period, okay, which leads to early seed production. The next question is, which hormone promotes internode and petiole elongation in deep water rice? Okay, so this is done by which phytohormone? Again, this is a very straightforward question. Ethylene. Okay, so ethylene helps or enhances the internode or petiole elongation in the deep water rice. And what does, okay, it helps to, okay, ethylene, we are talking about ethylene. It helps the regions of the, or you can say upper regions of the shoot to remain above water. Okay. So gibberellic acid, okay, is not correct. Okay, because it has some other roles in seed germination, breaking seed dominance. Kinetin, which is a cytokinin again, it has some other role, delaying the senescence and cell division. 2,4-D. This is a synthetic auxin. Alright, so these auxins roles are, what are the roles? Some of the major roles: root initiation and all, parthenocarpy, it promotes apical dominance. So answer is option C, ethylene. Okay, so this is the correct answer. So this was the end of this session. We will continue with the rest of the questions in the next lecture. Thank you very much for listening.